Hi everyone. This is the fourth and last of the formal lectures that I will give for the biofuels module. And you'll see this is again a journal club article. In the previous lecture, we introduced a lot of the biotech type of research that is being done in the biofuels field. And in this lecture, we'll look at this article, which is a review on some of the biotech work done in the yeast Saccharomyces cerevisiae, both to enable it to grow on a broader range of sugars, which is important for utilizing all of lignocellulosic biomass, and also in producing products that are not just ethanol, but products that might be interesting in a biorefinery concept. So this article is called Value-Added Biotransformation of Cellulosic Sugars by Engineered Saccharomyces cerevisiae. It's published in 2018 in a journal called Bioresource Technology, which has quite a high impact in this field, and it is generally, I would consider, a very strong article. So, by way of introduction, economic and sustainable implementation of large-scale lignocellulosic ethanol as an industry has yet to be realized. We've mentioned this before. There are some ethanol production facilities that use lignocellulosic biomass as feedstock that are online, but they make up a very small percentage of the overall ethanol production in the world. Many biofuel companies now focus on a wider range of products, which they call value-added bioproducts. So it is this concept where you will produce a large volume, low value product, such as your fuel of interest, bioethanol, if you're using lignocellulosic biomass and yeast, but also a range of other products which might be of higher value, and it allows you to address market demands and to make your overall business a little bit more economically viable and sustainable over a longer period. So just a reminder again that the majority of lignocellulosic biomass consists of cellulose, which is a glucose polymer, hemicellulose, which is a heteropolymer of glucose, xylose, arabinose, and other sugars, mainly xylose and arabinose, and also lignin, which is a polyphenol which cannot be fermented. And the composition of lignocellulosic materials differs quite a lot, as we've mentioned. This little table here just compares some important feedstocks for ethanol production in a second generation way. Miscanthus is a grass which can be used as an energy crop. And then we have energy cane, sugar cane, switchgrass, and cornstover here, and you'll see they all differ in terms of their composition of cellulose and hemicellulose, and that has a big influence on all of your downstream processing, your pre-treatment methods, your enzymes that are required to break them down, etc. If we look just at the monosaccharide composition, this also basically translates into different types of monosaccharides. If we were to hydrolyze all of the cellulose and hemicellulose in these different feedstocks, we're going to end up with different concentrations of glucose, xylose, and so on. But you will also see that we can't just concentrate on the utilization of glucose. Xylose is a large component in any of these feedstocks, and we need to be able to optimally utilize xylose as well. And in some of these cases, arabinose is quite a large amount, or present in quite a large amount as well, and we need to be able to utilize that arabinose to make our overall process economically feasible. So the lignocellulosic bioconversion process can broadly be broken into hydrolysis, which is where you turn the lignocellulosic biomass into monomeric sugars, and then the fermentation of that hydrolysate into a desired product. Efficient hydrolysis processes are still being optimized. As we've mentioned, there's a lot of work that needs to be done in pre-treatment, in enzyme production, and in optimizing the enzyme cocktails. But these include also acid-based, enzymatic, thermal, mechanical processes, and this is a multifaceted combination of approaches that are actually required in order to get optimal release of monomeric sugars from the cellulosic biomass. Merging the depolymerization and fermentation steps of lignocellulosic bioconversion has also been proposed. We've spoken previously about different configurations, being the separate hydrolysis and fermentation, the SSF process, the SCF process, or that CBP, the consolidated bioprocessing. Efforts have thus been put towards engineering yeast to efficiently consume all the lignocellulosic sugars, to produce a variety of products, and to resist the fermentation inhibitors present in hydrolysates. That kind of summarizes the main challenges for Saccharomyces cerevisiae as a CBP organism, or as an organism to be used in a biorefinery. We have to first get it to consume all the available sugars in the lignocellulose. It's very good at using glucose and doesn't use the pentose sugars, xylose and arabinose at all. Then we have to get it to produce a variety of products. It is very good at making high titers of ethanol to be used as a fuel, but it is not good at making a different type of product of higher value that we can also use. And then we have to get it to resist fermentation inhibitors. So we have to increase the yeast's robustness. We have to increase its ability to grow in that relatively toxic fermentation environment where you will have pre-treatment inhibitors present. So the first step then is to look at the consumption of all of the sugars in the lignocellulosic biomass, and we're going to look briefly at the consumption of xylose. As we said, xylose, normally the 5-carbon sugar that is present in the highest concentration in different types of lignocellulosic biomass. And there are 2 ways that have been found over the last approximately 3 decades that people have engineered into yeast in order to get it to use xylose. The first is as an isomerase pathway, and the other is called an oxidoreductive pathway. Those are both potential routes. They both have their advantages and their disadvantages. Looking first at the isomerase pathway, the enzyme is called xylose isomerase, and it takes xylose that is already inside the yeast and it isomerizes it to xylulose. And these xylose isomerases from fungal origin have been found to work the best. For a long time, this is basically the bacterial route of using xylose, and for a long time people tried to clone xylose isomerase encoding genes from different types of bacteria, and none of them worked well in yeast, and that's why people started developing the oxidoreductase path as well. About 15 years ago, there was a successful description of a xylose isomerase from a fungal origin, actually came out of a ruminant fungus, and that isomerase was able to turn xylose into xylulose at quite high rate. The reason we were still interested in the isomerases after the successful use of the oxidoreductase pathway will become clear. The oxidoreductive pathway, it first takes xylose and then reduces it to xylitol through an enzyme called xylose reductase, and that is followed by an oxidation step where xylitol is turned into xylulose by xylitol dehydrogenase. And it was found that the genes from the yeast Pichia stipittis, now called Shevazomyces stipittis, actually works the best for this process. But there's a problem here. As you see, I've indicated here the reductase prefers to use NADPH for this reaction, and the dehydrogenase prefers or requires NAD. So the reductase creates free NADP, and the dehydrogenase actually requires NAD and not NADP. And so what you get here is you get this cofactor imbalance. You avoid that cofactor imbalance with xylose isomerase, which is why that is preferred. But both pathways, either if you're using just the xylose isomerase or the reductase and dehydrogenase pathway, create D-xylulose, and that is something that the yeast can actually use. The yeast has a xylulose kinase to turn it into xylulose 5-phosphate. And that xylulose 5-phosphate is actually part of the pentose phosphate pathway. It's one of the intermediates of the pentose phosphate pathway. And in the pentose phosphate pathway, it can be turned into glyceraldehyde 3-phosphate or fructose 6-phosphate. Those are intermediates of the glycolytic pathway. And of course, then that can, through those glycolysis reactions, be used for energy or to produce ethanol. So this problem with the oxidoreductive pathway, that it requires 2 different cofactors, causes a cofactor imbalance. and that leads to inefficient xylose assimilation and accumulation of xylitol. So you get good production of xylitol until you get to a point where there's a lot of free NADP and no free NAD, and then there's no cofactor for the dehydrogenase to work with, and your metabolic pathway arrests here at xylitol, and so you get this accumulation of xylitol. And of course, if xylitol is the product that you want to make, and we'll discuss that a little bit later on, then that's great, but you actually normally want to utilize the xylose to make downstream products, either your fuel or a different product, as we shall see. So the cofactor imbalance can be harnessed to drive useful reactions, such as the NADH requiring acetate consumption pathway. That's a detoxification pathway. If you have a lot of acetate in your lignocellulosic hydrolysate, this can be quite useful, but that is not always the case. It depends on what your substrate is and what your pretreatment method is. So studies comparing the isomerase and the oxidoreductase xylose consumption methods have reported that the strains employing the oxidoreductase pathway actually consume xylose much faster. So in terms of gram per liter per hour amounts, your rate of xylose use is faster, but you turn some of that xylose into xylitol. So you will see that the strains that actually use the isomerase pathway will use xylose more slowly, but will yield higher titers of ethanol in the end. So it's still actually a trade-off between the two. And you still regularly see publication of both types of strategies being used. Yeast can also be engineered to use arabinose, the second most abundant 5-carbon sugar in lignocellulosic material. And again, it is either a bacterial isomerase that is used or an oxidoreductase pathway where the genes originate from fungi. So the E. coli ARA-BAD operon, which encodes an isomerase, a kinase, and an epimerase, will turn your intracellular arabinose into ribulose, ribulose 5-phosphate, and xylulose 5-phosphate, which again is part of the pentose phosphate pathway. The Bacillus licheniformis ARA-A was found to actually improve functionality. It expresses better in yeast than the E. coli gene, and then you get better utilization of the arabinose. The fungal oxidoreductive pathway uses an aldose reductase, a ribitol dehydrogenase, and then a xylulose reductase over here. And again, that then produces xylitol, and then xylitol dehydrogenase that you would also have to express produces D-xylose, which the kinase can then turn into xylulose 5-phosphate, and that enters the pentose phosphate pathway. A slightly more involved pathway, but the advantage is because these are fungal genes, they quite often tend to express at higher levels in yeast and work better in that case. However, you also see that there is again a cofactor imbalance. The use of the oxidoreductase pathway will lead to a surplus production of NADH. There's 2 steps that actually produces NADH and only 1 step that produces NAD, so you run into these cofactor imbalances again. And if you work in an anaerobic environment, you get to a part where you have xylitol accumulating and the process overall either slowing down or stopping completely. The second type of engineering of substrate consumption in yeast focuses on trying to express cellulases in yeast, so for consolidated bioprocessing, where we aim to decrease the requirement of these depolymerization enzymes in biomass before fermentation. We want to express cellulases. So that would enable the yeast to assimilate cellulose, and we require the expression of these cellulases so that we can get the monomeric sugar release from cellulose. There are 3 kind of ways of doing this as well. The way that we've most often used in my research group is kind of the simplest solution, I guess. That is the secretion of cellulose extracellularly to digest cellulose and glucose. And this kind of mimics the way that these fungal enzymes, which most often these enzymes are from fungal origin, and that is the way they are secreted in nature. So we simply produce the enzymes inside the cell, and they've got a secretion signal. They get secreted out of the cell, where they can then utilize the cellulosic substrate. This could there is a drawback here in that we have relatively low levels of cellulase secretion, and that's one of the points of research that we have in our group as well, is to try and different methods of getting the yeast to increase the amount of cellulase that it actually secretes. The second method that has been tried, that I've also mentioned previously, is the surface display method. Here the cellulase is secreted from the cell, but it's got a fusion to some kind of cell wall anchor. So the cellulase gets secreted, but then anchored on the cell wall. It stays attached to the cell wall and interacts with the cellulose or the substrate there. Releases the sugar, which can then be directly taken up into the cell. That's quite a big advantage. The drawback here, though, is the metabolic burden of these types of enzymes on the cell, and also, as I mentioned earlier, the processive enzymes, the cellular biohydrolases that you might want to make in this process, may actually not work terribly well, as these enzymes need to move along the glucose chains that make up cellulose in order to work properly. The third option is the expression of cellodextrin transporters that are coupled with intracellular hydrolysis to allow the release of these cellodextrins into free sugars. So what you have there is that they've expressed cellodextrin transporters from a cellulolytic fungus called Neurospora crassa, and that functioned probably when it expressed in yeast. So what do these cellodextrin transporters do? A cellodextrin would be a short chain of cellulose that is a product of the breakdown of cellulose. So maybe this is a cellobiose, 2 sugar units, and then we also call these cello oligomers, where you have 3 glucose residues, or 4 or 5, and they also recall, as you see, their cellodextrins. So cellodextrin transporters would bring these short chains into the cell. So following the intracellular transport, you also express at the same time an enzyme that will break down these cellodextrins. This can be one of 2 types of enzymes. You can get intracellular depolymerization by a beta-glucosidase, which is the enzyme I've mentioned earlier that hydrolyzes the beta-1,4 linkages and simply release glucose from the cellodextrin or from cellobiose in a hydrolysis type of reaction. And the other option is a cellodextrin phosphorylase, which doesn't use a hydrolytic reaction but a phosphorylytic reaction, which means a phosphate gets added to the sugar as it's broken down. And this could have significant energetic benefits, as you actually gain more ATP out of the complete hydrolysis or complete degradation of a phosphorylated glucose than you get from a unphosphorylated glucose. So that is one aspect of improving the substrate utilization of yeast. But after the depolymerization and hydrolysis of lignocellulosic biomass, you still have mixtures of hexoses and pentoses. And what you will often see if you grow yeast or bacteria on a mix of sugars, you get these kinds of strange-looking growth curves. Because of glucose repression, or catabolite repression, it is also called, of alternative carbon sources, these engineered yeasts will generally consume glucose before any other carbon source, and you get what is called dioxic growth. And so that's what we see here. This is just an example that I've added. It's dioxic growth. We get the typical S-type growth curve for the organism growing on glucose. And then following a short lag phase, you get a second little growth curve where the other sugar is used. So we don't get a co-utilization of both sugars, and that is ideally what we would want. So metabolic engineering strategies have been developed to enable simultaneous uptake of all available carbon sources, as this would increase the rate at which your product is formed and increase perhaps the yield of your product as well. And so we have to get yeast to use all of the sugars available at the same time, or pretty close to the same time at least. So glucose repression of yeast GAL genes can be eliminated by deletion of the XHK2 that encodes for a key regulator, and it allows simultaneous uptake of glucose and galactose. So that is something that has been found. Of course, we now need to find ways of simultaneously using the sugars that are actually available in lignocellulosic biomass. So enabling the simultaneous uptake of these two sugars has been very challenging in the last decade, and determination of sugar transporter kinetics indicated that xylose uptake was very strongly inhibited by the presence of glucose due to the competition for transport. So what they found is that actually yeast doesn't naturally use xylose, but it can still take xylose up inside the cell, and it does so through its glucose transporters. However, those transporters are far more efficient to transport glucose, and also when glucose is present, it actually inhibits the use or their use as a xylose transporter. So the kinetic inhibition of xylose transport that we see necessitates the development of a sugar transporter capable of simultaneously transporting glucose and xylose, or alternatively getting a transporter that is unique to xylose transport, so that we can express this in a yeast as well, so that it has a mechanism of both using xylose and glucose and getting both of those inside the cell. It's found that a conserved protein motif in the yeast sugar transporters played a very important role in determining its sugar preference, in determining whether it wants glucose or xylose, which at molecular level actually doesn't look that different from each other. And it was found that modifying this motif enhanced the preference for xylose, and the ability to transport glucose could actually be removed. They've also looked at transporters from several other fungal species. They've expressed them and mutated them in yeast, and they've shown a lot of promise. However, it's very difficult to find a transporter that is unique for xylose transport, that doesn't get inhibited by glucose, and that also works at a very low or that also has a very high affinity for xylose, because they have found some transporters that work for xylose only, but you need very high xylose concentrations. So they've got a high Km and a low affinity for that substrate. So as stated, salodextrin transporters in yeast enabled import and consumption of those salodextrins or salodextrin oligomers without complete depolymerization of cellulose to glucose. So that is what we said with the salodextrin transport. You can bring these salobios and salodextrins inside the cell. You don't have to get them down to glucose first. And so what they actually found was they made an engineered yeast that could use xylose, and then they also engineered this salodextrin transport into that yeast. And they found that co-fermentation of xylose and salobios could be demonstrated by expressing that salodextrin transporter in yeast that was engineered for xylose utilization. And what was interesting is that strategy actually avoids the inhibition of xylose uptake by glucose, because you're not taking up glucose into the cell, you're taking up a salodextrin. So the other transporters that transport glucose normally could now be used for xylose transport. However, the overall consumption rate and product formation rate wasn't up to industry standard yet, but this is a very interesting area of research. Okay, I'm going to stop this first part of the lecture here. We've now covered in quite a bit of detail different methods of trying to optimize yeast to use all of the sugars available in lignocellulosic biomass. And in the second part of this lecture, we're going to look at the production of some interesting products other than fuel ethanol that can be produced by yeast in a biorefinery setup.